Okay, so hello, sir.
Hi, Derek.
First off, what is your name?
My name is Brian Davis.
Oh,
I'm a
Dr. Brian
doc.
Davis.
I-
We have to make sure
Brian
we.
Davis, PhD. How about that?
There we
That
go.
works.
There we go. Uh, where did you go to school?
I spent my entire life in Texas, pretty much. My entire schooling is from Texas. All my university degrees are from Texas A&M University, which is where I got my PhD in the College of Veterinary Medicine.
Is that the gig them one? Is it
That's the gig'em. It's gig'em.
gig
Yes,
them?
gig'em.
It's gig them.
Yes. Wear my ring. Will forever wear my ring.
Sweet. Where do you currently work?
I am a geneticist at the National Institutes of Health in the National Human Genome Research Institute.
Ooh,
So basically, what that means is I built genomes. I built genomes for humans, for cancers, for dogs, for cats,
wow.
for exotics as well.
That's pretty amazing. That's pretty amazing. Well, I
I'm very
appreciate you being a part of this new thing that I'm trying out, and you get to be a part of history, Dr.
don't.
Davis.
It's what we all
Um,
want to be.
which
Some of us
yeah.
want.
So basically, and I'm gonna there just a couple moments to kind of tell you about this because this is gonna set the tone for what it is that I'm doing here. I'm doing a new thing on my YouTube channel, and it is an interview type segment, and I'm gonna call it the conservation equation. Okay. Now, basically, the way that the whole thing sets up is global conservation efforts. They're this massively complex thing. Okay. And a lot of folks, such as myself, uh, uh, you could say that I know a couple different things, about like big cats and their behavior, and how they think, but there's so many other forces at play when you're talking about total global conservation efforts. There's political factors, there's geographic factors, there's climate, there's biologic factors, there's sociologic factors, and it gets very big and it gets very confusing, which is why me, as a not expert of these things, I want to talk to experts such as yourself, to hopefully shed a little bit of light on some of these individual little components of this overall equation. Eh?
Sounds
Eh?
fantastic. Yeah.
And see, I mean,
Education, entertainment.
I'm actually wearing a sweater that is like more of like a collegiate kind of thing to make myself look smarter.
I see. You have the collar. I see.
I do. I clean up. I shave. It's great. So we're gonna make this whole we're gonna make this thing work. So thank you
Sounds
very
great.
much for being a part of this whole thing. Now, I would just like to ask you, just what is your work comprised of?
Primarily, my work at the National Institutes of Health is looking at the population structure of dog breeds, so domesticated dogs. Also looking at disease characteristics like cancer in dogs, cancer in humans, as well as domestic cats. Now, this is moving forward into other species. So, exotic animals is really where we're trying to build my personal effort, my collaborators' effort towards, which is why we are setting up and have set up a very large bank for tissues and genomes for exotic animals that are in captivity within the United States.
And
So
that's how you and I,
it's
came to know of each other's existence.
very true. I came begging for genomes, as it were, and talking to you about our efforts a
Please,
couple
sir,
years
can I
ago.
have some genomes?
Please, I'd like some. I have many now. In the past few years since we've met, we have an amazing bank so far, but it's only still a small slice
Hmm.
of the population of. We focused on cats to begin with, exotic cats to begin with. Although we do collect genomes from many other animals, we haven't really delved into reptiles or amphibians. here.
No, 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 no,
No,
no,
ice and snow for sure.
no, no. Okay,
Okay.
so I am back to recording. I apologize for that. Dum Dum forgot to register his Skype recording software.
Well, I'm
So yeah.
fine that you're experimenting on me, getting it all
Yeah,
set.
I know. I got to do that. My scientific method and my trials and errors and everything. I'm still wearing my sweater, so I mean, at
Yeah,
least people can, think like, oh, he probably knows what he's doing. So that's a good
hey,
thing.
you convinced me. I'm convinced.
Okay, so the last thing that we were talking about was the repository.
Yeah,
Was the genetic samplings that you are in the process of trying to attain. So we can talk a little bit.
we basically what we're doing is we're collaborating with the exotic reserves, the small zoos, larger zoos as well in the U.S. to simply record the genomes of the animals that they are husbanding.
Hmm.
Or they no longer exist. I get emails every week about animals that are passing,
Oh wow.
and if they're not sampled, there's no real record except for on paper their medical records that they existed. But the genome, their genome is irreplaceable. It's unique, right? So if we can sample the genomes of these animals, we will forever have a permanent, indelible record that these animals existed
Sure.
for whatever purposes someone wants to use for them. If people want to go and study the population of tigers in captivity, we can do that now. We can do that in 10 years from now
Oh
and
wow.
see how the population has changed. There's innumerable reasons to want to study these populations, but we can't do so if we don't sample them.
So how long does it take to do like one individual sequence? Like if you were to get one sample of one of our cats, like let's say Conopoli
Sure.
out in the compound,
Yeah.
and then from the time that you actually open up that sample, whether it be a blood sample or a tissue sample or a cheek swab sample, how long does it take you generally from receiving it to actually having a sequence of that animal?
So if we're going to sequence the whole genome of an animal, we would want to do it from blood because the amount of DNA that you get from blood, or in the case of perhaps a euthanasia, a tissue or of some
Sure,
sort.
sure, sure.
So cheek swabs we can do a little bit of small testing on, but we wouldn't want to really sequence the genome, at least not with technology now. In the future, it's entirely possible that we would be able to do that. But the time frame is about 6 months. It's under 6 months
Okay.
to do that if we start immediately.
Sure,
But the
sure,
key
sure.
that I want to emphasize is that we are really pushing towards the sampling first, right? We're not sequencing every animal that we receive, not yet at least, right? We're trying to capture them, and we have cryopreservation. So we will freeze their tissues, we will freeze their DNA and it's good forever.
Now does that have to do with like the our capacity for computation? Um, like the time frame?
That
Because
that is part of it, but mostly it's cost prohibitive at this point.
okay, okay,
Uh, it's still $5,000 to do a good quality genome, and that's just not something that we are able to do right now.
like
To do a
15,
whole
15
genome,
years ago, like 15 years ago, what would have been the time scale and the cost?
it wasn't feasible. It
Yeah.
just wasn't. I mean, the Human Genome Project in '03, it was hundreds of millions of dollars. It was both a private and a public effort.
Wow.
So the cost is just plummeted. I mean, it's amazing what technology has allowed us to do. It's still not within a price point that you or I are gonna, spit in a cup and sequence our whole genome. It's
But
getting there.
hundreds of
But we
millions,
want to.
hundreds of millions funneled down to 5,000. That's pretty good
I
progress so
know.
far.
I'm very, very pleased to be a genomicist at this point right
Okay.
now. It would have been a lot more costly to be a genomicist in the early 2000s for certain.
Okay.
But we are with the Exotic Genome Project, we're actually capitalizing on a lot of tools that were developed in the Human Genome Project,
Sure.
and as people are sequencing other species as well. So there are tons of tools that are available to use. It's becoming a lot more easy and computationally feasible to assemble genomes and to create a brand new genome that never existed before.
Oh,
which
wow.
people are starting to do for cancers and things like that. That's one of the efforts that we're trying to do is make a genome for a specific cancer. They're doing it in humans, moving on to dogs, and we would like to do so for the tissues that we capture for exotic animals.
So are you like no, not doing full? You know, you say you're not doing like full genomic sequences, but you're kind of looking at these little, like
As of
some of these little
correct,
pieces right now. Like what are
as of right now, yeah.
sure, and like a lot of work as far as cancer research is concerned. Um,
So for cancer research, we kind of do need whole genomes. That's
sure.
for to study cancers, we need because not only do small little parts of the genome change, they flip around. Chromosomes break and rejoin with other chromosomes. Sometimes they shatter. So we need that whole genome to study cancer, and
Sure.
we're working towards that right now. But we're just not there yet. We're still in the sampling phase. You know, I know that we've been discussing this for a couple years now, but
Mm,
it's taken a while to get the word out and to get the facilities to store all of our samples. We've established a database with a registry. We're in collaboration with a large group of people, including Conservators Center,
oh
to
wow.
have unique identifiers
I've seen some of
for
those
all
guys.
those.
Yeah, on my
They're
social
great.
media stuff. They've been
Yeah.
doing some things. I applaud their efforts.
Yes, all of this gets done as a result of collaboration. We can't do it
Absolutely.
alone. I'm just one researcher who spends way too much time working.
Mm,
So I have a network of scientists that are working with us, and obviously people like yourself and like Mindy Stender, the Conservator Center, and Doc Antle. And yes, yeah, without you guys, we simply wouldn't exist.
well, yeah,
But
and I definitely want to bang the drum for this cause because I think it's a tremendously important kind of thing. Well, and here's the thing: when we're talking about again global conservation efforts, like
yes,
what my whole goal is here on the conservation equation. Um, Basically, we may not necessarily like in what you were saying. We may not necessarily possess the tools to be able to gain the insight that we need to know, like right now. But we probably more than likely
sir.
will have such tools in
There
the
there
relatively
are
near future.
there are tools that do exist to do that right now. We are doing it, but just not on a whole genome level. We
Well, and
take little bits
what I'm
as you
yeah,
alluded to.
and what I'm saying is like as time goes on and more people are going to have access to the information and things become less cost prohibitive, it's going to be imperative now that we build up these samples that
Yeah,
we
we just need to sample them.
yeah
That's it.
yeah
That's our goal right now. There are research efforts. We're looking at population structure, for instance, within the captive population of multiple cat species. We are doing some whole genome efforts, but those are really kind of secondary. Those are kind of proof of concept. We really need to do the sampling first, above all other things. And once we have a sample, it's a record.
so
It's a permanent record.
what are some of the insights? I mean, and I know that it's still like a lot of this program and a lot of the things that you're doing are still like in the relative infancy. But already, what are some of the insights that you folks have already kind of stumbled upon just from the work so far?
Sure.
Is it stuff that you don't want to have to? I understand. Is this
So
stuff
there
that you're still
are we
working
have a
on? Is
number
stuff
of
that you don't want to
publications
have
that are
published yet?
forthcoming. We have many publications that are forthcoming.
Of course.
Can't tip my hat. I can't tip my hand just yet.
I
I
know.
will say though that
I'm sitting there trying to just like we got the cards and everything, and
yes, right.
it's like
No, I
looking at
want
them
everyone
and
to know. I want everyone to know, but not before it's a scientific
no, you gotta
publication.
have to. Yeah, you gotta get all this. And yeah, look. I'm like the Hollywood Reporter, being like, so what's the scoop on genomics? Huh?
Well,
Tell us.
there are a few things, but they're relatively obvious things.
Sure.
For instance, when we look at populations of animals in the wild,
Hmm.
as their populations get smaller, they become more and more inbred and less genetically diverse.
Sure.
Well, when we look at a slice of the captive populations in the U.S. and I call them American populations because
Sure.
they are American populations. They are sampled from all over the world, but they are American populations. They're actually significantly more, okay, they are more genetically diverse.
Interesting.
than the populations in the wild.
Now,
Um,
and
we
what?
can talk
Sorry
about
to.
if that's good for conservation or bad for conservation, but for the species, for preserving the species, there's really no question that a more diverse species is better for a population.
That is definitely an interesting kind of thing. Now, when you're talking about a lot of the captive populations here in America, there's been so much interbreeding between like the different subspecies, the
Sure.
Sumatrans and the Bengals and the
Absolutely.
Siberians and whatnot, and it's almost like it's kind of created this melting pot mixing, of tigers. Now, that's another thing I would love to be able to talk to maybe like a conservation biologist who could actually then make the determination of whether or not like a homogenized version of a subspecies could actually have conservation viability, which is like the only reason we have that question now is because seemingly the work that you guys are kind of spearheading.
Well, we are not advocating for any specific conservation in the wild plan
Hmm.
right now. We're really focusing on the captive
Sure.
population. Whether or not there
But you don't have to.
are
You don't. I
local
can do
adaptations,
all
but
that.
it's no, it's entirely possible that there are local adaptations for subspecies. I mean, they've been documented in the past that just over a few thousand years, these populations, they'll become maybe thinner, more easy to maneuver in their where they live. They may have immune adaptations,
Sure.
uh, metabolic adaptations. It's certainly that they've adapted to their niche, but we don't really appreciably know if it would be better to have a heterogeneous species population, rather than this extremely small subspecies population.
Hmm.
And that's not a question that we're seeking to really ask, at least not at this point.
Sure.
But we certainly think it's an interesting question that other scientists could use our data for, to help inform them on their hypotheses.
See, you hear that? Other scientists. That's what we're trying to figure out. We're learning things, and then now we're coming full circle, asking questions. Conservation equation.
I'm always for the math. I
There we go.
definitely am all about the math. Fantastic.
So you gotta bring it around full circle.
Got
Gotta.
yes.
This is really fascinating stuff, and we have and I certainly feel like we have an aging population of big cats just in the country in captivity. I think that I believe that a lot of the breeding percentages have gone down more in recent years, um, and that almost kind of creates this sense of expeditiousness about this type of project because as those aging populations continue to die off, it's going to definitely make the work that you're trying to do, that much more important. I believe. So definitely, any other what like where are where's a good place that people can go who would be in situations like mine, like with they take care of animals, of cats. Where would be a good place for them to go in order to possibly contact you or come into contact with you, and then you know put
Sure.
yeah put their genetics into the what's the word I'm looking for?
We have a
The
repository.
repository.
That's what we like to
Oh
call
my gosh!
it. Yeah.
Yeah.
It's
See,
yeah.
I got my sweater on. Sweater.
Yeah. I couldn't help but use a large word like repository. It's a science brain, you
This
know.
is a science thing.
So so we dubbed it the Exotic Genome Project. There are other places that are sequencing exotic animals, but it's really a captive exotic repository. So we have a website, exoticgenome.org. It's a very basic website. We are not web designers, but there's a contact form on there. There's also an intake form. What we're able to do is we can send you sample kits for tissues. If your animal has cancer, we can sample the tumor tissue when the tumor is excised. We can do cryopreserve this tissue for future study, as well as, and this is becoming more popular, as you mentioned, the aging population, we have a tissue panel kit. So basically, when the agreement is with the husbanders, when an animal is to be euthanized, when their quality of life has decreased, we have a tissue kit that we send out. So we will actually record the gene expression in all of their major organs. And it's through this diverse sampling that we can actually kind of get a genomic topology, like a basic understanding of how the genetics of each of these species exist. These are things that are just not possible to do in wild populations, right? So, and by using this information, we can compare animals that are healthy or towards the end of their life with animals that have a specific disease. So if an animal has a cancer susceptibility, their progeny have a cancer susceptibility, hopefully we can start to tease apart those genetic risk factors and understand the mechanisms of cancer or disease and how it relates to both humans and companion animal populations, like domestic cats, domestic dogs.
Sure, that's pretty incredible. And the fact that you're able to, take a lot of the information that we're gleaning from this exotic population, and then you're actually able to apply it not only to exotic populations, but also to people populations is pretty incredible stuff. Now, as far as these kits are concerned, now these are kits that pretty much any veterinarian can use in the performance of like a necropsy,
absolutely,
type thing.
It's a simple media, and all it really does is preserve the RNA. So if we remember basic genetics, RNA is gene expression. It says how much of every gene in the entire genome is on at any specific point in any tissue, and that varies with the tissue. It even varies within a tissue, but we're kind of starting at the organ level.
Sure.
So,
and you said exoticgenome.org, right?
.org. Yeah, exoticgenome.org. We had a more complicated.
This one. Oh, is that backwards?
That's it.
Is that backwards?
That's absolutely it.
Can you read that?
It's correct.
Oh,
you got it.
I see it as backwards. That's weird. This is my right. This is my left. That's weird. Okay, never mind. I'm not going to try to.
Are you sure
technology. Oh my goodness, how do these things work? Skype and whatnot. It's like, I don't know. I'm like a parrot. You put a blanket over my head. I think it's night.
Hey, I started life as a computer science geek. I've been a computer nerd my whole life. That's why I love genomes.
Yeah.
It's data to me.
But
I do not want to mess with tech support on Skype and all that kind of. I'm done. I'm done with that. I'm going to handle the genomes, the biology, and other people can handle computers. Thank you.
That is pretty. And as far as like right now, you're laying the foundation for.
That's really what it is.
This work now. What are some of like the big really ambitious, large scale, like way off in the future? What would you love to see accomplished through the foundation of this work eventually in the near future, or even in the far future as well?
So the key is to really establish an American centralized genomic database for everyone to have access to. Now, let me just to clarify, just because you submit a sample doesn't mean that you have to approve that someone else can use that genomic data. We want to record all of this information for each individual person. So for all of your animals, Derek, we want you to have the ability, free of charge, just to mention, there's no charge for any of this. We want you to be able to preserve the genome of every animal that you've had in your care, if you so choose. For everyone, we want that to be able to have access to it for everyone.
Yes.
But as far as scientific goals, my personal scientific goal is to tie the line between disease characteristics across species. Most of us are mammals, right? No, we all share a lot of common biology. There's a lot of common biology. And now that exotic animals, they have double the lifespan that they do in the wild. They're under immense veterinary surveillance as similar to companion animals like cats
Sure.
and dogs. And what we've seen in the past decade or so is that researchers like Leslie Lyons in cats, Steve O'Brien in cats, my boss Elaine Ostrander in dogs, there are definitive mechanisms that are shared between these species in how they develop disease.
Oh wow.
If it's whether it's cancer or whether it's optic disease, kidney disease, it's drawing a parallel between all of these different species and having a common treatment mechanism,
Sure.
right? It's a when we talk about being a good steward of the biology that's in this world, whether it be us, whether it be our fellow man, whether it be our companion dogs and cats, or whether it be exotic animals that are getting extinct, they're moving towards extinction in the
Sure.
wild, having this sort of integrated one health approach to treatment is really kind of the holy grail for health for health across mammalia. So that's my high hopes. If I can contribute to that in my career, I'm happy. And it starts with sampling. We've got to document these animals. We have to know they existed if we are going to examine them in the context of their health records, and how related they are to the rest of the population, and be able to compare them using these new genomic tools that are coming out all the time from places like where I work, NIH, and the Broad Institute at MIT. It's amazing. It's an exciting time to be a genomicist.
It certainly is. It certainly is, and it's almost like that you're. It's like taking this larger kind of top down approach to a lot of these ailments and
That's
disorders and diseases. And if you can knock out like this large chunk of ailments, and then we can knock out this other larger chunk, and then we can keep on just kind of whittling it down and making things a little bit more quality. I would imagine.
that that is certainly the way that I think that it's going to work.
My
It may
goal,
may take
my
decades,
oh
but
what
it's going to work.
my goal for your work is going to like armies of android tigers. Like the Fifth Element kind of thing, where like they basically had a couple like live cells like from the crash ship, and then they made Lilu like in that little
That's
pod
it's one of
thing.
my favorite movies. It's one
Oh
of my
yeah,
favorite
it's
movies.
awesome. Love that. That's basically what I think of like the stuff that you're doing, and I'm just like maybe someday they're gonna be able to just like like tuk, 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 and just like make a whole bunch of Kanapalys. I'm just like that's cool.
Hey,
And I know, and I'll make the disclaimer. I talked to you about that, and you've said to me so many times, like, look, I have to talk about things that are actually rooted in reality. So,
for now,
yeah,
rooted in reality for now.
but I can definitely take like the wow, gee whiz, Mister approach to a lot of this stuff. So that's okay. I'm totally imagining like we're just gonna make 3D printed tigers and just out into the wild someday. I think that would be amazing.
Well, when we talk about human genetics, all of the advances in human genetics, being able to print 3D organs, being able to make pancreas from stem cells, it all started with the Human Genome Project.
Yeah,
That's
absolutely.
how we were able to understand the basics. That is the ultimate basics. And though tiger genomes have been sequenced and other things like that, we need thousands of genomes to
Sure,
really understand
sure.
what makes up a species and how they are different. And so
Okay.
that's what we're shooting for, is the thousands of genomes that exist, tens of thousands of genomes across mammalia that exist just in felids and canids within the US. So it's all about that baseline, getting that infrastructure in place so that we can build tools, use existing tools to understand all these animals
Yeah.
that we care for.
So basically, what I want to do right now then is to talk to the individuals out there who follow my content and who also take care of big cats. You know who you are. Dr. Davis.
I may have spoken to you at some point.
Yeah, Dr. Davis has a tremendous program, a historic program that you could absolutely be a part of. The forever preservation of the animals that are in your care. So please, very, very much so, consider being a part of Dr. Davis's program. That's a really awesome thing. So It's up
I
to
do
that.
I do want to emphasize they can send me an email at NIH as well.
Awesome.
Exotic Genome is just a very basic website.
What
We're
is your
working with ad
email? What
content.
is your email?
My email is Davis BW at mail.nih.gov. Davis BW at mail.nih.gov. Nailed it.
Sweet.
Nailed it.
Okay,
Yep.
I'm gonna keep it up here just so people have it. Or they can just do a
So
screenshot. Never mind.
that's the
21st century
quickest way to
technology
send me an email.
results. Or you know what? I gotta put this into an editing program anyway. So I mean, it's not like I can't just kind of on the bottom. I don't know why I do these things. It's just silly. Just like I gotta write something down and just hold it up to the
There
camera.
you go.
That's the only way we can solve this problem.
Low-tech solution.
It's so silly. I'm just like this. You're a scientist. You work at the National Institute of Health, and you guys are like, hold on, let me write this down. So yeah, it's silly. Okay.
Well, I'm sure you
Um,
got it.
I think that's probably a good stopping point for the
Okay.
conversation. Is there anything else that you can think of that you would like to talk on
Sure.
this platform, good sir?
Well, it's through efforts like this that we will be able to integrate science. So let me just, as an example, one of my friends has started a project to record the vocalizations of exotic cats. It's called the Pruston Project, prustonproject.org.
Hmm.
Basically, what they are doing is they're looking at hybrid cats, they're looking at species of cats, subspecies of cats, and actually trying to document all of their vocalizations and analyze them in a similar fashion
Oh wow.
that people analyze birds. Well, I say there's a genomic component to it, and I think they agree, but we need genomics and we need people recording these other aspects of life. I wouldn't have thought of that. I wouldn't have thought to
Wow.
go and record vocalizations. I'm just happy, hearing the snow leopard yowls and
Yeah.
tiger chuffs. I just like that. But establishing this genomic tool set that other people can query at will, I mean, it's really going to forward science, and I'm just excited to be part of it.
Yeah. Well, and that's the great thing when you have that it's almost like you're creating this library. This
It is a
library of
library,
genetics.
without a doubt. That is the most accurate term for it.
And
It is
these other
a library.
individuals with their unique perspective can go into the library, and then all other new avenues of information and insight can then be opened. And
That is
that's very
completely the
exciting.
case. From tiger striping pattern to skull shape to vocalization pattern to parts of their genome that expand and contract, to disease susceptibility, to the gene expression in every tissue in their body. All of this information, we just want to record it. We just want to record it, and we want intelligent people to come up with questions that they can ask, and hopefully we can help them.
That's so cool. That is so awesome. Alrighty, Dr. Davis, I appreciate you being a part of the flagship episode of my new YouTube interview series called The Conservation Equation. I think I have to do that now every single time.
It's
This is
integration. That's integration,
yeah,
mathematical concept.
that's right. That's right. Because it's equation and mathematics, and we're talking about and like these are the different bits of information of science, like science and things and whatnot. We're coming together. Conservation equation. Awesome. Thank you
Hey,
so much, Dr. Davis, for
no,
being a part of
thank
this.
you for what you do. I
All
mean,
right.
without what you and Heidi do and your friends, I tell you, it's way harder than my job. I sit at a computer and I think and I talk and I write and read.
Oh,
So you,
Pisha.
thanks, Derek.
Thank you, sir. All right.